Hi everyone, good afternoon and welcome to today's webinar. I'm so glad that you could join us today. My name is Xiaoxuan and I'm the Marketing Executive at Biocytogen Pharmaceuticals Beijing Company Limited. I'll be your moderator for today's session. I'm joined by some amazing colleagues from the Biocytogen team who will be our panelists today during the Q&A session. So we have Yihe Chen and Shiyu Zhang, who are our BD managers for the APAC team. So Shiyu is fluent in Japanese. So for our Japanese speaking attendees, please feel free to submit your question in Japanese as well. Thank you both for being here today. We are excited to have you on the panel. Now, it's my pleasure to introduce today's speaker, Dr. Meiqi Zhang. Dr. Zhang earned her Ph.D. at Peking University, where she focused on the mechanism of copper chaperone proteins and the use of copper chaperone antagonists in tumor metal immunology. She then joined Biocytogen as a product manager in the BioMice Product Department. Dr. Zhang leads the development of humanized mouse models for neurological disorders, as well as models that support large molecule drug penetration across the blood-brain barrier. Her work has enabled a wide range of humanized mouse models to be applied across multiple therapeutic areas, including Alzheimer's disease, Parkinson's disease, ALS, epilepsy, and other rare diseases. So in today's session, Dr. Zhang will cover the structure and function of the blood-brain barrier, strategies to enable drug penetration across the BBB, key clinical evaluation methods for receptor-mediated transcytosis or RMT, and examples of relevant target humanized models such as TFR1, CD98HC, and IGF-1R. So before we get started, just a quick reminder, the Q&A box is already open. Please feel free to send in your questions at any time. We will save the time at the end to answer as many as we can. So with that, let's get started. Dr. Zhang, over to you.
Yes. Okay, thank you, Xiaoxuan. And hello, everyone. My name is Meiqi, and I'm a product manager from Biocytogen BioMice Department. And today I will share animal models developed by our company for blood-brain barrier crossing. And these models can aid the preclinical research. The BioMice department is one of our company's pipelines, and we can provide preclinical products and services. And based on the gene editing services, we have developed over 4,000 of the shell humanized and immune deficiency animals and cell models. And together with the pharmacology services, we can provide the one-stop customized project plans. And now all of the top 10 MNC are using BioMice. And we have a large population of the product lines. We have over 1,000 target humanized mouse models, including the immune checkpoint humanized mice, cytokine and cytokine receptor humanized mice, and other humanized mice such as the GPCR humanized mice. And we have about 100 seriously immune deficiency models. And we also have many other disease models, including in the aging, in blood disease, in the metabolic disease, and in some neurological disease. And we also have about 700 gene knockout mouse models, and these models can be used in the target validation. And this is today's condense. And first, I will take a brief introduction about the structure and the function of the blood-brain barrier. And next, I will mainly introduce the key preclinical evaluation methods for the blood-brain barrier penetration using the receptor-mediated transcytosis. And the last one is the summary part. And first, the blood-brain barrier, the structure and the function. Blood-brain barrier, which is named the 3B, is a highly specialized interface that takes control of the entrance of the molecules in the vascular compartment to the brain. And the BBB protects the CNS system from toxins and pathogens while regulating the exchange of the essential nutrients and signaling molecules. And it said that more than 98% of the small molecular drugs and about 100% of the larger molecular drugs, including the antibodies, enzymes, and some nucleic acid-based drugs, cannot cross the blood-brain barrier because of its structure limitations. And to address this restrictive nature of the blood-brain barrier, multiple strategies have been developed. And one of these is by the receptor-mediated transcytosis, which is named the RMT strategy. This strategy is the most sociologically relevant approach. So next, I will introduce this strategy. The receptor-mediated transcytosis strategy is a cellular transport process that it can help in the movement of specific molecules across the blood-brain barrier, and it uses some receptors based on and highly expressed on the blood-brain barrier according to its function, including the endocytosis. It can transport the larger molecular drugs from the blood to the brain. And the most famous of the targets is TFR1, CD98 heavy chain, and IGF-1R. And so next, first I will introduce our humanized models for the TFR1. TFR1 is the receptor of the transferrin and functions in ion transport through its transcytosis mechanism. And this target is highly expressed on the brain endothelial cells. And so many methods have used the TFR1 in the penetration across the blood-brain barrier. So the most famous one is the platform developed by Denali. They use the TFR1 for the blood-brain penetration. And this is the targeting strategy of our humanized TFR1 mice. We humanized the TFR1, we humanized the extracellular domain of the TFR1 and remained the transmembrane and intracellular domain as the mouse sequence. And we also do some validation data towards this humanized mice. And the first is the mRNA expression analysis, and we can see that the mouse TFR1 mRNA was knocked out in the homozygous mice. And in this mice, it can only express the human TFR1 mRNA, and this sequence was also confirmed by Sanger sequencing. And we also perform protein expression analysis in bone marrow, and we can also see that mouse TFR1 cannot be detected, and only the human TFR1 can be detected. And we also detected 3 female mice and 3 male mice, and we can see that the TFR1 expression remains consistent across the different gender. And we also have a protein expression analysis of TFR1 in the brain endothelial cells by flow cytometry. And we can see that the mouse TFR1 was only detected in the wild type mice, but not in the humanized mice. And the humanized TFR1 can only be detected in the humanized mice. And in the wild type mice and in the humanized mice, the TFR1 expression in the 2 mice remained consistent. And we also detected protein expression in multiple tissues, including the heart, liver, spleen, lung, kidney, stomach, colon, muscle, and brain. We can see that TFR1 can be detected in all of these tissues. And in the expression levels of the TFR1 in the wild type mice and in the homozygous mice remained consistent. And here is also the protein expression data in reproductive organs, and we can see the similar results. And next, we detect the healthy status of these mice. And first, we perform the leukocyte cell populations. And we can see that humanized TFR1 cannot change the overall frequency or distribution of immune cells in spleen, blood, and lymph node. We detected many immune cells, including the TB, NK cells, and some T cell subpopulations. And here is the blood routine test and biochemistry test. Since TFR1 function is related with iron transport and related with red blood cells. So if TFR1 doesn't function, the mice will have anemia. So we detect 20 parameters in the blood test, including the red blood cell count, the HGB, and 2 parameters of the red cell distribution-wise and reticulocyte. These parameters are mainly used in the assessment of anemia, and we can see that all of these parameters remains the same as that in the wild type mice, indicating that these mice don't have anemia. And we also detect 3 parameters related with iron homeostasis, including the serum iron, mouse ferritin, and mouse transferritin. We can also see that in the humanized mice, these 3 parameters remains consistent with that in the wild type mice. So this data showed that TFR1 can function well in these humanized mice. And next, we do some applications of these mice. And first, this is an imaging method to see if antibodies can enter the brain. We use the 2 antibodies targeting the TFR1 and conjugated with fluorescence module, and we can see that the antibody can enter the brain, and the fluorescence signal in this group is higher than that in the isotype group. But this is a low resolution method that can't accurately assess whether the drug is still on the vascular blood wall or the drug have actually entered the brain parenchyma. So we use another high resolution method, which is IVIS, to image the antibodies in the brain. We also conjugated the antibodies with a fluorescence module, and we can see that the signal was enriched in the blood vessels, but we can also see signals in the brain parenchyma. And this experiment can prove that the TFR1 antibody can actually enter into the brain parenchyma. And besides the imaging methods, we also have ELISA methods assessing the pharmacokinetics of the antibodies. Here is the workflow, and we can collect the blood sample, and if needed, we can collect the CSF. And after the Herter perfusion, we can collect the brain. And usually we can take half of the brain to do HC staining, staining the anti-IgG, and we can take another half of the brain in the ELISA detection. And the brain tissues was homogenized, and by adding the dextran, we can separate the solutions into 3 layers. And the top one and the middle one is the solutions of the brain parenchyma, and the bottom one is the solutions containing the capillaries. And we only use the top and middle one as the we only use the top and middle one in the ELISA detection to get accurate antibody concentrations in the brain parenchyma. And our internal experiments showed that the concentrations in the whole brain and in the parenchyma can be 5 to 6 times different. And using this method, we have detected many antibodies PK study, and in this study, we use the GR141 analog as a positive control, and this drug is a positive drug that have been launched. And we also have another 4 antibodies named antibody 10, 14, 15, and 16. These 4 antibodies were developed by our company, and we can see that both each TFR1 antibody can enter into the brain parenchyma, and under the same concentrations, our antibody 16 have a higher parenchyma penetration rate compared with the positive control. And many other clients have also used our mice to do some PK study, and this is one of the case of our clients. And he just developed an anti-human TFR1 bispecific antibodies and do the PK study, and we can see that the TFR1 bispecific antibody have a higher brain serum ratio compared to the control group. And this is another application case developed by one of our clients, and they developed many antibodies with different affinities, and we can see that as the affinity decreases, the antibody concentration in the brain parenchyma first increase and then decrease. This is also consistent with the theoretical basis because if the affinity to the TFR1, if the antibody affinity to the TFR1 is too strong, it will make it difficult for the antibody to dissociate, causing it to be retained in the blood vessels and making a lower parenchyma penetration rate. And besides antibody drugs, TFR1 can also be used in AAV capsid penetration into the blood-brain barrier. And this is a literature published by one of our clients on Science. And they developed a reprogrammed AAV capsid and conjugated with a TFR1 antibody. Using TFR1, they can take the AAV capsid penetrate into the blood-brain barrier. And on June of this year, FDA have approached this AAV capsid for safety research using our humanized TFR1 mice. Besides brain, TFR1 can also be used for drugs delivery into the muscle. And here is a double humanized mice developed by Biocytogen, which is the TFR1 and DMPK double humanized mice. And in this mice, we test the inhibitory efficiency of the antibody oligonucleotide conjugates drug, which is AUC drug, against the human DMPK in these mice. And we can see the MRI detection results that DMPK was knocked down in the muscle tissues. And this proved that TFR1 can used in the drugs delivery into the muscle. And here is also another literature published by one of our clients, and they are using TFR1 for the drug delivery into the muscle. And here is the summary of our TFR1 mice. Our TFR1 mice, the knock-in strategy of this mice is full genomic in situ replacement of the entire extracellular domain. And we have tested many validation data of these mice, including MRI expression data, protein expression data, and some other function function assay data. And these mice, the phenotypes of these mice is normal, including from our blood biochemistry and some other analysis and serum area data, birth data, and gyroscope data. And now we have validated these mice can be used in the monoclonal antibodies or bispecific antibodies, AOC drugs, and AAV capsids penetrating into the brain, or this can also can be used in the drug delivery into the muscles. And this mice is one of our most popular mice, and these mice have monthly sales of over 1,000. And we in the world, more than 15 customers have used these mice. And besides the single humanized mice, we also have many double or triple humanized mice crossing with the humanized mice for some drug delivery research. And next, I want to introduce the humanized models for CD19 heavy chain developed by Biocytogen. CD98 heavy chain is a multifunctional protein involved in amino acid transport and cell signaling, and it can form a complex with the light chain like L81 to transport large neutral amino acid. And CD98 is highly expressed on the blood-brain barrier, and it can also be used for the brain delivery of some large molecular drugs. And this is also the transport platform developed by Denali. And for this mice, the targeting strategy is a little bit like the TF1 humanized mice. We only humanized the actual domain of the CD98 heavy chain, and the transmembrane and the intracellular membrane was remained mouse sequence. And we also perform some validation data of these mice. First is the mRNA expression analysis. We can see that the mouse mRNA cannot be detected in the homozygous mice, but only the human CD98, but only the human CD98 mRNA can be detected in the homozygous mice. And this sequence is also confirmed by Sanger sequencing. And then we perform the protein expression analysis in the brain microvessels. We can see here that mouse CD90 heavy chain can be only be detected in the wild type mice, but not in the homozygous mice. And the human CD98 heavy chain can only be detected in the homozygous mice. And we can see that CD98 heavy chain is highly expressed in the brain microvessels with an expression rate close to 100%. And we also detected multiple tissues expression of the CD98 heavy chain, including heart, liver, spleen, lung, kidney, and brain. We can see that CD98 heavy chain is highly expressed in the brain and the kidney, and low expressed in the liver, spleen, and lung. And we think the advantage of CD98 heavy chain over TF1 is that the CD98 only highly expressed in the brain and shade and kidney, and maybe the peripheral delivery efficiency may be lower of the CD98 heavy chain. And here is the immunofluorescence staining of CD19 heavy chain in the brain, and we use the mouse CD31 as a brain muscular cells marker, and we can see that the human CD98 heavy chain can expressed in the brain cells. And next, we also have some data detection the health status of the mice. And the first is also the leukocyte cell subpopulations analysis. And we detected many immune cells in the spleen, blood, and immune node. We can see that humanized CD98 heavy chain doesn't change the overall frequency or distribution of immune cells. And here is also the blood routine test and the biochemistry test. Both each parameter from the two tests was the same in the humanized mice compared to the wild type mice, indicating that CD98 heavy chain humanized mice can function well. And last, we also perform a PK study using this single humanized mice, and we can see that the anti-CD90 heavy chain antibody have a high brain serum ratio compared to the control IgG. So we want to say which target is better between the TF1 and the CD98 heavy chain, and what are the difference in their efficiency in penetrating the blood-brain barrier. So we crossed the TF1 humanized mice and the CD98 heavy chain humanized mice to make a double humanized mice. And in this double humanized mice, we can compare the anti-TFR1 antibody and the anti-CD19 heavy chain antibody. And we can see that in these experiments, we also use GR141 as a positive control of anti-TFR1 antibody, and we use CD19 4B as an anti-CD19 CD98HC antibody. And we can see that anti-TFR1 antibody exhibits a higher brain exposure in the 24 hours after dose, but anti-CD98HC antibody exhibits higher brain exposure in the 72 hours after dose. And the metabolic rate of the CD19 heavy chain antibodies is very slow. So this double humanized mice can be used for the comparison of the penetration efficiency of the two shuttle molecules. And these experiments maybe can provide some insights into whether we can choose TFR1 or CD19 heavy chain. And the third target is IGF-1R. IGF-1R is a tyrosine kinase that can bind to IGF-1 or IGF-2, promoting cell growth, survival, and metabolism. And this receptor is also highly expressed on the blood-brain barrier. And targeting IGF-1R may be a promising strategy for delivering larger molecular drugs into the blood-brain barrier. And this is a platform developed by ABL. They use the IGF-1R in the penetration of the blood-brain barrier. And for this humanized mice, our gene strategy is also the extracellular domain humanization, and the transmembrane and intracellular domain was remained mouse sequence. And here is some validation of this humanized mice. The left one is the mRNA expression analysis. We can see that the mouse IGF-1R mRNA was knocked out in the humanized mice. And in this mice, we can only detect the human IGF-1R sequence. And this sequence was also confirmed by single sequencing. And the right one is the protein expression analysis. We also detect IGF-1R expression in multiple tissues, including heart, liver, spleen, lung, kidney, brain, and colon. And we can see that IGF-1R can be detected in the brain and the lung in high expression levels, and in heart, spleen, and colon in lower expression levels. And here is also an immune fluorescence analysis of the IGF-1R in brain vessels. And we also use mouse CD31 as a blood vessels marker. And we can see that human IGF-1R is highly expressed in the blood vessels. And here is the function assay of this mice. Our design scheme is under the human IGF-1 addition. It will activate the receptor, and the receptor phosphorylation will increase. But if we add a positive drug targeting to the human IGF-1R, like the fidgetumab, maybe the phosphorylation of the receptor will be blocked. So we performed these experiments, and we can see a very good results that under the human IGF-1 addition, the phosphorylation of IGF-1R increased, but this phosphorylation can be blocked by the addition of the antibody. These experiments proved that humanized IGF-1R can function well in these mice. And here is also some detection data about the health status of these mice. And this is the leukocyte subpopulations analysis. We detect many immune cells in the spleen, blood, and lymph node, and we can see that the humanized HF1R doesn't change the overall frequency or distribution of immune cell types. And here is the body weight growth curve of this mice, and it can grows normally, and the body weight growth curve is consistent with that of the wild type mice. And the last part is the summary. Now the most popular strategy for crossing blood-brain barrier is by the receptor-mediated transcytosis, using some receptors based on or expressed on the blood-brain barrier. And the most widely used the targets is the TFR1. And the second or the third one maybe the CD198 heavy chain and IGF-1R. Both the three targets we have developed many mice models. For the TFR1 related mice models, we have single humanized models on the B6 background, BioMice background, and two immune deficiency background. And we also have humanized rats. And the ligand of TFR1 is TF, so we also have the double humanized mice. And based on this single humanized mice, we also crossed these mice with many other mice models or disease models. First is some neurodegenerative disease models such as the AD and the PD. We have TFR1 and tau double humanized mice, and we also have crossed the TFR1 humanized mice with the 5XFAD mice. And for the PD, for the Parkinson disease model, we have crossed the humanized SNCA with the human TFR1 mice. And besides besides the brain, TFR1 can also be used in the drug delivery into the muscle. So we also crossed the TFR1 humanized mice with many neuromuscular or muscle-related diseases such as the DMD, DM1, CMT, and some targets in losing weight and gaining muscle. We also have the double humanized mice. And for the CD98 heavy chain related mouse models, we also have single humanized models in the B6 background, BioMice background, and immune deficiency background. And we also have humanized CD19 HA heavy chain rats. And these mice, we also crossed these mice with many other disease models, like in the Alzheimer's disease. So we have double humanized mice, and we also have human CD98 heavy chain plus with the 5XFAD mice. We also have this double humanized mice. And for the 1GF1R, we also have a list of mice models, and we have the first version and the plus version, and we also have its ligand humanized mice like IGF1, and we also have the double humanized mice. And last one is we also have many humanized models for AAV receptors. We can use for some AAV capsid delivery. And here is all my sharing. And next is the Q&A session. If you have any questions, please feel free to ask.
All right. Thank you, Meiqi, for the excellent presentation. I can see we already have some questions coming in through the Q&A box. So let's go ahead and take a look at those. So and to everyone in the audience, if you haven't submitted your question yet, there is still time. And so please feel free to send them now. So just a quick reminder, if you prefer to ask your question in Japanese, you are welcome to do so. We have a panelist here who is Shiyu, who can respond in Japanese. Shiyu, maybe you can talk a little bit.
皆様、こんにちは。すいません。ごめんなさい。本日のウェビナーご参加いただき誠にありがとうございます。で、私はバイオサイトでの、臨床線の CRO サービス事業部の、BD の長と申します。本日のウェビナーについては、日本語でご質問可能になりますので、ご遠慮なく Q&A ボックスでご入力ください。はい。よろしくお願いします。ちょっとなんか、日本語の質問がありますので、こちらでまず、ちょっと答えておきますね。で、佐藤様からちょっと質問がありまして、人化後 TFR1 の正常に機能するのか、また、マウスのトランスフィレントの交差反応性はどうなるのかという質問ですね。でこちらについては、ちょっと簡単に説明いたしますで、まずは、機能性については、その、人化 TFR1 で、人化のマウス体内で成長に機能することを、弊社でもう試験されましたので、それを確実で確認されています。で、その根拠としては、その TFR1 の接口系のノックアウトマウスでは、重度の貧血や、胎児摂取神経障害など、ちょっと起こすことがありますので、その文献も報告されています。しかし、そのヘテロの接口体でも、その異常な、その赤血球やその組織に鉄の欠乏が派生されていることもあります。でも、弊社で搾取したそのTFR1 もヒトカマウスなんですけど、その血液学およびその血液性化学の検査については、もう健康な状態を維持することで、もう証明されましたので、特にその血清の鉄のレベルとか、野生型と比べたら同じでした。はい、以上です。
So, thank you, Sri, for the question and answer. So let's take another question. So we have here is, do you have validation data for the delivery of small nucleic acids? Meiqi, maybe you can answer that.
Yes, we have many application cases of this, and we have validated small nucleic acid drugs delivery into the brain using our TFR1 and Tau double humanized mice, and we also have small nucleic acid drugs delivery into the muscle using our TFR1 and DMPK double humanized mice.
I'm sorry.
I think here is another question. Could you clarify the difference between the humanized IGF-1 mice and the plus mice, particularly in terms of genetic design and their applications in the blood-brain transport studies? And for the first version, we use the WPRE module in the 3' UTR, and for the plus mice, we use its original 3' UTR. So the difference of these two mice is the protein expression levels. The protein expression levels in the first version is higher than the plus version, and it's exceed the normal expression levels. So we developed the plus version, and now we are validating the blood-brain transport efficiency using the plus mice. And we haven't get this data, and maybe in next 2 to 3 months we will get this data.
Okay, thank you, Meiqi. So maybe we take a look at another question. Let's see. So, do you have mice that combine an Alzheimer's disease model, 5XFAD and TFR1?
Yes, we have these models, and we also have some validation of the strain. Not only the 5XFAD mice, but also the TFR1 crossed with this mice, the double humanized mice. We also have some validation data, and this data including some phenotypic data such as A-beta deposition, astrocyte activation, and some biomarker detection. And we also have behavior tests such as water miss. And this screen is available now. So if you have any question, you can or you are interested in the lines, you can contact with our BD.
Okay, thank you. So while waiting for more questions to be submitted in the Q&A box, so maybe our one of our BD manager from the APAC team, Yi He Chen, maybe she can share a bit more about like what are the possible questions that customer or clients would ask when they deal with us. So Yi He, maybe you can share a little bit more.
So it's very nice to have you here. My name is Yi He Chen, and I'm responsible for the Japanese market. And there's a lot of clients ask me questions. For example, is there some difference between the females and the males and TFR1 locking mice? So Dr. Meiqi, could you answer this question?
Yes, we have such validation data, and we detected protein expression in the bone marrow in both the female and male mice, and we don't see any difference between the female and the male mice. And we also have detected the mRNA expression in the female and male mice, and also we don't see any difference in the two mice. But if you want to use TFR1 mice in some neurological disease models such as 5XFAD mice, the 5XFAD mice will have gender difference in its phenotype in these phenotypes. But for the TFR1 humanized mice, there is no difference between the female and the males.
Thank you for your answer. And there actually for the practical issue, sometimes for the international transportation, we generally recommend using the transport the females because they usually like more milder and they don't fight with each other. So generally, if you don't have any preference for the gender, then we recommend you to choose the males.
Yes.
So if you have any question, you can just directly put your question in the question box, and we can answer it directly.
I think there is another question. Is there also data on brain distribution in wild type and homozygous mice using antibodies specific to human TFR1 that do not cross-react in mice? Actually, in the application cases that I today shared, all the antibodies used were species specific to the human TFR1. They will not cross-react in the mice. So in the former study, we will use both the wild type and homozygous mice in the PK study, and later we will only use the homozygous mice. And if you are interested in the data, and I think we can collect the information and reply to you later.
So is there any question that you'd like to answer, or shall we? All right. Thank you for the excellent questions. So I think we have had some really great discussions today. So I'm afraid we are coming to the
Xiaoxuan,
end of our... Oh, yeah.
Maybe
You wanna
there
take
is
it?
another question.
Okay, sure, no problem.
Yes. Is any data related to the compression for efficiency in aspects of penetration of BBB among 3 transporters on BBB? And for now, we only have TFR1 and CD98 heavy chain double humanized mice. So we have comparison data between the TFR1 and the CD98 heavy chain. And for the IGF-1R, we are doing the PK study, and we haven't get the data now. Maybe in the future we can compare the three transporters in the blood-brain barrier penetration rate. Yes, that's all. Okay.
Okay, thank you. So I think we just need to wrap up the Q&A session, and I mean it's coming to the end of our session here today. So a big thank you to Meiqi, Shiyu, and also Yihe for sharing your insights. Thank you our audience for attending this presentation as well. If you miss any part of today's session, this webinar will be available on demand. You can request access or catch the recording anytime from our website or on our YouTube channel. If you have any follow-up questions or you would like to have more information about our models and services, feel free to reach us through our website at biocytogen.com, or you can connect us on LinkedIn as well. So we will be happy to continue the conversation. All right, I think that's all for us today. So thank you again for joining, and we hope to see you at our next webinar. Thank you and goodbye.